Randomized controlled trials are considered the gold standard design to infer causality. However, RCTs are expensive, time-consuming, and often unfeasible to conduct. Example, because of poor long-term compliance and ethical issues about random treatment allocation. Thus, relationships of modifiable risk factors with cardiovascular disease events have mostly been investigated using observational study designs, such as case control and cohort studies, which cannot reliably infer causality, as confounding and reverse causation bias can distort the finding. The tool of multivariable Mendelian randomization analyses allows us to get a little bit closer to predicting causality. These can be long-term studies. Before this, it's very difficult to cover, or pretty much not feasible at all to have a randomized controlled trial across many decades. That's going to be still fairly well controlled. And to me, they're kind of a combination, or they have similar traits to observational studies and randomized controlled trials that kind of add to the context and allow us to have greater certainty by making up for some of these limitations. When it's isolated, it's associated with increased risk of atherosclerotic cardiovascular diseases. But when we account for ApoB, we see that the reason LDL-C is associated with these outcomes is because LDL-C is associated with ApoB. And when we factor in ApoB, we no longer see the association. But when we do the inverse, we still see an association, suggesting that ApoB, even when considering LDL-C, is the causal factor for atherosclerotic cardiovascular disease. So I guess that's a good place to start in that. Hey everybody, welcome to episode number 4 of the Talking Studies series, where I break down interesting research papers that I've read and share some kind of cool and useful findings that you might be interested in. Today we're going to cover a study called Mendelian Randomization for Cardiovascular Diseases, Principles and Applications. And although it talks about cardiovascular diseases in the title, this is mainly just a review on the background of Mendelian randomization in general, which you may be wondering what exactly that is. And I guess that's the whole point of this video, because this is a new study design, or at least relatively new study design, that I think if you're like me, you've been hearing a little bit more in kind of the health and fitness spheres, especially as it pertains to cardiovascular disease. And it has some kind of interesting applications and fills in some holes from or that are left behind by other study designs. So I guess that's a good place to start, in that if you followed along at all, I forget what it's called, but I have a series of blogs that I've been writing, kind of breaking down different study designs. So I talked about animal models, I think I talked about in vitro models, I talked about observational studies and randomized controlled trials. And within each of those blog posts, I talked about the pros and cons of each of those designs. And observational studies, for starters, kind of has some pretty commonly touted limitations, in that observational studies essentially consist of tracking or just observing outcomes in individuals over time. You're not using an actual intervention or experimenting with anything. You're just gathering data, for example, a bunch of demographic data, like whether or not people smoke, what type of activities they do, what type of foods they eat, and then over time tracking to see if those people end up getting certain diseases or at what rates they are dying at and things like that. This is useful in some regards, but some key limitations are that observational studies are or generally tend to have less accurate measurements. So as opposed to like a 4-week trial in a laboratory where you could track, or researchers can track everything that subjects are eating and everything they're doing, in an observational study, commonly the researchers might just send out a survey or questionnaire asking what subjects have been eating for the last month or the last year, and they might only send these kind of food records or questionnaires out once every year, or maybe once every few months, or once every 6 months. So although there's some validity to the data they're collecting, it's not quite as accurate, likely, as data you'd be collecting in shorter, more experimental trials. Another issue is lack of control. So as I kind of alluded to, in randomized controlled trials, you can control all of the extra variables, all of the things that the subjects are doing or eating, for example, and really limit the changes or the differences between the two groups, the intervention group and the control group, to just the variable of interest. For example, if you wanted to look at the effect of how much saturated fat or artificial sweeteners one group, or the effect of consuming those foods on a given outcome, you could heavily control all the other variables so that both groups are eating the same exact thing, other than that one difference or that one change that you're making, in this case regarding saturated fat or artificial sweetener consumption. You don't have that type of control in observational studies because people are free living, and you're not experimenting or limiting anything in particular. You're just kind of tracking and observing things as they happen. And this kind of goes hand in hand with the last key limitation I kind of have listed here, which is vulnerable to confounding. Since you're not controlling for all of those things, you may wind up in a situation where there's an association between X food and X outcome, or X macronutrient and X outcome. But in reality, people who are more likely to consume X food group or X macronutrient are also more likely to do Y, and that Y variable is influencing the outcome. Now, RCTs can make up, or randomized controlled trials, I'll abbreviate to RCTs sometimes, can make up for some of these issues in that they're more heavily controlled. These are actual experimental studies where, like I described, researchers are going in and controlling variables between two groups and really trying to narrow down just one variable of interest that is being manipulated. But they have their own vulnerabilities and their own limitations as well. First off, commonly they're not free living, or they can tend to be less ecologically valid. What does this mean? Essentially, in a research or laboratory setting, individuals might be behaving differently than they would in real life settings. And even outside of that, um, there can be a difference in terms of efficaciousness and effectiveness, or a question of efficaciousness versus effectiveness. In that, sure, if you are have subjects in a study where nutritionists are calling them up once a week to remind them to adhere to a given diet, and they actually do adhere to that diet, then maybe they'll get the end goal or the outcome that is shown in the study. But the question remains of whether or not free living individuals who aren't getting those calls from nutritionists are going to actually be able to make those changes to achieve those outcomes. And in general, like if you look at exercise science research, for example, sometimes they might be comparing one calf exercise to another calf exercise, and they're limiting the subjects so that the subjects aren't allowed to train the rest of their body for the rest of the week. And this isn't ecologically valid, or I guess realistic, in terms of how people actually train. So when people go to apply these results, it's uncertain whether or not the results from just doing a given calf exercise in the context of not doing any other exercises is going to have the same result as doing that particular calf raise exercise, but in the context of also training the other body parts of your body for the rest of the week. Another limitation, or a couple of limitations, just have to do with practicality. So randomized controlled trials tend to be more complex, more difficult to run, because you have to control for all of these variables. You have to get individuals to do these very particular things or follow these protocols. And for this reason, they also tend to be more expensive, right? A lot of times, uh, subjects will be rewarded or incentivized with some type of monetary payment, or they're given meals for free to help them kind of adhere to the protocol. And that makes these studies more expensive as compared to just sending out an online survey, or just recruiting individuals for an observational study. Another limitation is that, going along with this kind of practicality aspect, since these are more complex studies and they're harder to run, typically they will have smaller sample sizes or smaller amount of subjects in them to account for this, to make it easier for researchers to track all of these individuals. And also, sometimes it's just a matter of difficulty in recruiting subjects to follow maybe a particularly strict dietary protocol over 6 months or a year. They can only get so many people to sign up, and so the sample size is small. And so then there's questions of whether or not the results are due to just only having a few dozen subjects, and also whether or not those results will be more generalizable to the remaining 7 billion or whatever it is people in the world. And also, these randomized controlled trials tend to be shorter, because for all these practicality reasons I just described. Just the same reason that it might be difficult to get people to join the studies, it will be more difficult to have people adhere to the protocols in the studies over longer periods of time. So you see that these studies will typically be in a matter of weeks or months rather than over years or decades. And just like RCTs can fill the holes of limitations to observational studies can fill some of the holes to RCTs, and back and forth. Now, Mendelian randomization trials can come in and additionally fill some of the holes. And to me, they're kind of a combination, or they have similar traits to observational studies and randomized controlled trials that kind of add to the context and allow us to have greater certainty by making up for some of these limitations. Quote, randomized controlled trials are considered the gold standard design to infer causality. However, RCTs are expensive, time consuming, and often unfeasible to conduct. Example, because of poor long term compliance and ethical issues about random treatment allocation. Thus, relationships of modifiable risk factors with cardiovascular disease events have mostly been investigated using observational study designs, such as case control and cohort studies, which cannot reliably infer causality as confounding and reverse causation bias can distort the findings, end quote. So, just like I kind of alluded to, randomized controlled trials and observational studies have their limitations that kind of leave holes unfilled in terms of answering questions to things like cardiovascular disease, and particularly to things like drug treatments, which might not be as easy to test due to ethical reasons. If you think that a drug might have particular adverse effects, it might take a while to go through many, many trials to try to figure out whether or not it's actually safe to even test whether or not the drug works. And then also for lifestyle interventions, there are considerable gaps that are left unfilled, even when combining RCTs and observational data. And here is where Mendelian randomization comes in. I guess first I'll explain what exactly Mendelian randomization is. And the first place to start, I think, is explaining who Mendel is. So this study design is named after a guy named Gregor Mendel, who was a geneticist. I don't know how many years ago. I don't have the timeline perfectly laid out, but he is really well known. He's like kind of considered the father of genetics, I believe, for three theories he came up with in particular. Three laws. There's the law of dominance, which pretty much describes how when we're looking at genetics, you have a given gene that is the instructions for a given protein. And there are different variations in that gene, right? Different mutations that can cause that protein to look a little bit different one way or another based on the instructions of how to build that protein and which amino acids to fill in which place. And so you'll get a different gene or a different allele that's like a different copy, a different version of that gene for that protein from your mom and from your dad. So you get one from each. You can kind of think of these like recipes. They both might be recipes for chicken parm, but they might be tweaked a little bit so that one has a little bit more sauce, one has a little bit more breading, etc. And you get one each from your parents. The idea of dominance is that one of these genes will show through more than another. So if we simplify it down to like pea plants, which Gregor Mendel was looking at when he came to the conclusions of these three laws, you might have a gene that impacts height, and it might be dominant, meaning that if you have the dominant allele, so the version of that gene that causes the plant to be tall, just having one allele will cause the plant to be tall. So even if that plant inherits one tall allele, one genetic mutation that causes it to be taller, and one that causes it to be shorter, it will end up being taller because that gene is dominant. So that gene, or that allele is dominant, I should say. That allele will show through. Um, in the same instance, if you have two tall alleles, the plant will be tall. But if you have two short alleles, then the plant will end up being short. So I'm not sure I explained that super well. I'm sure some geneticists are listening and kind of cringing at my lack of nuance, but that's the law of dominance kind of in a nutshell. It's maybe the least important for the laws that pertain to Mendelian randomization. The second law is the law of segregation, which um, basically describes how you get one allele from each of your parents. So during the kind of reproductive process at a cellular level, when the egg and the sperm are mixing to form a gamete, you'll get one allele from each parent for every gene. Now, the third law is the law of independent assortment, which says that which alleles you get are is random. So your dad has two alleles for a given gene. Your mom has two alleles for a given gene. We'll call that gene one. And then they also each have two alleles for gene two. And the law of independent assortment says that just because you get allele A for gene one from your dad doesn't mean you're going to get allele A for gene two from your dad. You might get allele A from both, or you might get allele B from both, or you might get A and B or B and A, so to speak, if that makes sense. But it's random. So it's not like uh, you have all of your father's chromosomes lined up and they get split down the middle, and you get all of the alleles that are cut on the left side. It's random. So you might get some on the left side or right side from a bunch of different genes. Again, not sure I explained that with perfect clarity, but maybe this quote will clear it up. I got this from another paper called Genetics Autosomal Dominant. I'll share that or leave the link below. Um, Quote, first, the law of dominance and uniformity states that some alleles, which are variants of a particular gene found at the same chromosomal locus or location, are dominant over other alleles for a given gene. Those traits that are not dominant are termed recessive. If an organism inherits at least one dominant variant, then it will display the effect or phenotype of the dominant allele. Second, the law of segregation states that the two alleles for each gene separate from each other during gametogenesis so that the parent may only pass off one allele. Thus, the offspring can only inherit one allele from each parent. Third, the law of independent assortment states that the alleles of different genes segregate independently of one another during gametogenesis and are distributed independently of one another in the next generation. This concept was later verified with chromosomes, though also disproven in some instances. For example, when genes occur on the same chromosome, they can be linked and not follow this law. End quote. Yeah, so that is where the name Mendelian randomization comes from, and it'll make a little bit more sense in a second. The first step of a Mendelian randomization is actually acquiring data from what's called a genome-wide association study, which essentially gathers a bunch of genetic data from a bunch of people and associates certain genetic mutations or genetic variants, as they're also called, with outcomes. So if you have a bunch of data on all different kinds of people, individuals with a certain genetic variant might be more or less likely to have heart attacks, for example. Quote, genome-wide association study is a hypothesis-free study design that tests the association of thousands or millions of genetic variants with a phenotype. The principal aim of a GWAS is to identify variants that are associated with the phenotype, which can be used to identify genes that are relevant to the etiology of the phenotype or to develop a predictive polygenic score for the phenotype, end quote. So all in all, we're looking at a bunch of different genes, a bunch of different variants of those genes, and seeing which ones associate with different outcomes or different risk factors. Now, Mendelian randomization takes these two concepts. First is the GWAS, so that certain variants are associating with certain outcomes, and then Mendel's laws of segregation and independent assortment, which essentially act as randomization in a randomized controlled trial. So the authors explain this through the example of calcium and cardiovascular disease risk. So if we wanted to do a randomized controlled trial to test whether elevated calcium levels will lead to increased cardiovascular disease outcomes or events, we might have a bunch of people randomly split into two groups: a control group that takes a placebo pill that has nothing in it or nothing relevant in it, and another group that's the experimental group that takes a calcium pill for every day for like a year or whatever the extent of the trial is, and then we see which group is more likely to have cardiovascular disease events. Mendelian randomization essentially replicates this idea. It's just instead of giving them a pill, you have a bunch of people who are randomly assorted to either have higher or lower calcium levels based on genetic mutations. So just based on genetics, some individuals will happen to have very high calcium levels in their blood, and some individuals will happen to have very low calcium levels in their blood. We can look at these individuals across a lifetime and see whether or not individuals with higher or lower calcium levels will end up having more or less cardiovascular disease events, just like we would in a randomized controlled trial. Quote, the principle of Mendelian randomization refers to Mendel's second law of independent segregation of genetic alleles when DNA is transmitted from parents to offspring at gamete formation. This is similar to the random assignment of treatment in an RCT, which aims to produce groups with similar clinical characteristics, hence reducing the risk of confounding, end quote. And so in this way, Mendelian randomization can fill in some of the gaps that are left behind by randomized controlled trials and observational studies. First, that part I just explained where we have randomization, which you're missing in observational studies. And we also have control in that if done properly, or if we're looking at very useful genetic variants, the genetic variant will only affect the one variable of interest. So we're controlling all other factors. All other factors are randomized, right? The amount of individuals in group A and group B should randomly be distributed across different variables like smoking status, BMI, gender, sex, race, height. All of these things should be randomly distributed between these two groups. They should be relatively equal in these two things. The only thing that should really be different is whatever is impacted by the genetic variant that we're looking at. Also, we have the benefits of this study being carried out in free living conditions, right? We're not changing the way that these individuals are acting. All we're looking at is their genetics and their risk of certain outcomes over a lifetime. And that lifetime piece is the next advantage, in that these can be long term studies. Before this, it's very difficult to cover, or pretty much not feasible at all to have a randomized controlled trial across many decades. That's going to be, still fairly well controlled and in free living subjects. Or I guess in free living subjects is more likely in long term studies. But you get my point in that this allows for really long term studies, the way that observational research allows for studies to follow subjects across decades. And lastly, it's practically easier and faster than randomized controlled trials. Quote, Mendelian randomization studies have several advantages over RCTs. They are often faster and cheaper to conduct, as they can be conducted using existing large-scale GWAS data. Mendelian randomization studies can inform on potential causal relationships between modifiable risk factors and rare diseases that would require extensive sample sizes and long-term follow-up for sufficient endpoints to occur in an RCT. Moreover, Mendelian randomization studies can investigate exposures with expected adverse effects on disease risk, which would be unethical to test in trials. Randomized controlled trials for a modifiable risk factor or medical treatment usually examine short-term effects, as long-term compliance can be difficult to achieve and cost increases with longer duration. In contrast, as genetic variants are fixed at conception, Mendelian randomization results reflect the effects of lifelong perturbations in the risk factor. Thus, Mendelian randomization is a valuable study design to overcome several of the limitations and problems confronted in conventional observational studies and RCTs. Nonetheless, Mendelian randomization should not be considered as a panacea, as this design comes with its own set of assumptions and caveats as described below. End quote. And now we can kind of get into maybe some interesting nuances and applications when it comes to Mendelian randomization studies, now that you have kind of a background or base knowledge of what they are. So first, some requirements, concerns, and limitations. There are three particular requirements that the authors of this study outlined. First is that the variant at hand reliably associates with the risk factor of interest. So if the risk factor is elevated LDL cholesterol, we want to make sure that the variant reliably is associated with that risk factor and fairly strongly associated with that risk factor. Secondly, we want to make sure that the variant does not associate with confounders or other risk factors that influence the ultimate outcome. So if the ultimate outcome in this case would be cardiovascular disease, and we're assessing whether or not the variant is associated with LDL cholesterol, we want to make sure that the variant isn't also associated with blood pressure, another risk factor that is influencing cardiovascular disease. And lastly, we want to make sure that the variant does not influence the outcome in another causal pathway. Quote, the three core assumptions that must hold for valid results in a Mendelian randomization analysis are illustrated in Figure 2. Specifically, the genetic variant or multiple genetic variants used as instrumental variable for the risk factor must, 1, reliably associate with the risk factor under investigation, the relevance assumption. 2, not associate with any known or unknown confounding factors, independence assumption. And 3, influence the outcome only through the risk factor and not through any direct causal pathway, the exclusion restriction assumption, end quote. So if you look at Figure 2, which I'll throw up on the screen, in this example, they're looking at whether or not a genetic variant for alcohol intake influences cardiovascular or coronary heart disease outcomes. So they're pretty much testing actually whether or not alcohol over a lifetime is associated with these outcomes based on genetic variants that are associated with alcohol intake. Now, requirement number one is that the genetic variant must associate strongly with alcohol intake. It must influence alcohol intake, whether or not you have a certain genetic variant for the gene at hand. Second is that you want to make sure the genetic variant does not also influence your confounding variables like smoking. So you want to make sure that it doesn't also influence the likelihood or amount of smoking that individuals will do. And lastly, you want to make sure that the genetic variant isn't somehow also um, associated or linked to the outcome in via another causal pathway. So you want to make sure that the genetic variant itself doesn't also cause the outcome, in this case, coronary heart disease events. Jeez, I got to get some water. I'm getting dry from talking about all this. Okay, continuing on. Quote, the plausibility of the second assumption can be evaluated by examining whether the genetic variant is associated with competing risk factors. So in this example, we can explore whether or not the genetic variant is associated with things like smoking or BMI or etc. The third assumption cannot be assessed directly, but must be justified by biological knowledge. In Mendelian randomization analyses involving multiple genetic variants, the plausibility of the assumptions can also be assessed by statistical methods, end quote. So it appears the third requirement, the exclusion restriction assumption, that the variant at hand does not influence the outcome by some other direct causal pathway, is a little bit more difficult to ascertain and requires kind of another body of research, which is kind of mechanistic data or other data supporting biological plausibility. So just like Mendelian randomization fills holes for RCTs and observational studies, there are still holes that need to be kind of filled, or support that needs to be provided from other research to make Mendelian randomization research useful. Now, moving on to some concerns when it comes to interpreting this data or the validity of these types of studies. First is pleiotropy, which is essentially the idea that certain genetic variants can affect multiple different phenotypes or influence multiple different traits. Quote, pleiotropy refers to the association of a genetic variant with multiple phenotypes. Horizontal pleiotropy refers to the association of a genetic variant with more than one phenotype on discrete biological pathways. This pleiotropy is of concern as it violates the exclusion restriction assumption and can distort the results. Vertical pleiotropy refers to the association of a genetic variant with more than one phenotype on the same biological pathway, which does not invalidate the findings. If we move down, this is kind of demonstrated in figure 3, which I'll throw up on the screen. We can see here is for horizontal pleiotropy, for example, if we're trying to look at whether or not coffee consumption leads to coronary heart disease, it's important that the genetic variant doesn't influence smoking as well as coffee drinking, because smoking is another risk factor for coronary heart disease. So horizontal pleiotropy would be when the variant impacts multiple risk factors. Vertical pleiotropy, on the other hand, it involves the variant impacting one trait that impacts another risk factor that is directly causal to coronary heart disease in this example. So in this example, the variant would impact physical activity or how much individuals move, and that is not necessarily causal to coronary heart disease, at least in this example. Rather, that then influences body weight, which is then causal in this kind of diagram explanation to coronary heart disease. So since this genetic variant would impact several phenotypes or several kind of traits, in that it impacts physical activity, which then impacts body weight, which ultimately impacts the outcome at hand. Moving on with concerns is something called linkage disequilibrium, which entails several genetic variants being inherited kind of together or in similar places on chromosomes in your DNA, so that they get passed on together. Quote, linkage disequilibrium, which refers to the correlation of genetic variants in the population, is another potential caveat in Mendelian randomization studies. Genetic variants in physical proximity on the same chromosome can be in linkage disequilibrium. Confounding would result if the genetic variant used to proxy the risk factor of interest is in linkage disequilibrium, i.e., it's correlated with another genetic variant that is associated with the outcome through a pathway that does not involve the risk factor of interest, end quote. So, for example, if we have gene A and gene B are right next to each other on your DNA, they may tend to be inherited um, with each other. And so, what you'll see is that looking at genetic variants of gene A, we might think that as gene A is, let's say, associated with the risk factor of exercising, and gene B is associated with the risk factor of smoking, we might think that gene A is associated with the outcome of cardiovascular disease, and we might then go on to presume that exercise is somehow influencing cardiovascular disease, and in that more exercise might be leading to more cardiovascular disease. I'm just making this up as I go, when in reality, the association we see between that genetic variant associated with exercise and cardiovascular disease is really just because it's commonly inherited with gene B, which is associated with smoking, which is then associated with cardiovascular disease, if that makes sense at all. So, pretty much uh, an individual gene or gene variant could simply be associated, or an individual gene or gene variant that influences a given risk factor could suggest that that risk factor is associated with an outcome. But that might not actually be the case if there's this linkage disequilibrium where that genetic variant is commonly inherited with another genetic variant associated with a different risk factor that actually does influence the outcome at hand. Moving on with limitations, when we're looking at different genetic variants, they're not all created equal when it comes to predicting risk. And as a result, they're not all equal in terms of their applicability or usability in Mendelian randomization studies. Quote, genetic variants typically have a small effect on most risk factors, i.e., they explain a small proportion of the variation, which can lead to low statistical power in the Mendelian randomization analysis and the risk of false negative findings. For example, the fat mass and obesity associated gene, FTO, is the locus with the largest effect on BMI, but this locus explains less than 0.5% of the variation in BMI in populations of Europe ancestries and even less in populations of other ancestries. The corresponding variation explained by all near independent genetic variants, which is 941, found to be associated with BMI in a GWAS meta-analysis involving about 700,000 European ancestry individuals, was about 6%. The amount of variation explained by known genetic variants is often below 5% for complex phenotypes. End quote. So the issue being described here is that if a given risk factor is not very heavily influenced by genetics, then there will be a small influence of genetics on that risk factor, and therefore it'll be more difficult to ascertain based on these genetic studies whether or not that risk factor is associated with the outcome, because there'll be a very small signal that we're trying to pick up, since the variation in the risk factor according to individuals' genetics is very small. For example, someone on the really high spectrum for a given risk factor due to a genetic variant, let's say it's BMI, for example, as they're using, might only have a slightly increased risk of having a high BMI than someone who's all the way on the lower end in terms of genetic variance influence on that risk factor. So someone who has, let's say, the peak gene that influences or causes increased BMI, they might only have a slightly increased risk of having, an overweight BMI compared to someone with the gene on the other end of the spectrum that predisposes them to have a lower BMI. If that makes sense at all. I find myself saying that a lot with this genetic stuff. It would be kind of confusing to put into my own words, but I guess that's good practice for learning the material more. Moving on, a good example of the importance of how much in a given risk factor is influenced by genetic variants. Quote, observational findings of inverse association between physical activity and risk of major cardiovascular diseases have gained little support from Mendelian randomization studies. Suggestive evidence of strong inverse associations has been observed for genetically predicted vigorous physical activity and risk of myocardial infarction, and for moderate to vigorous physical activity and risk of subarachnoid hemorrhage. However, other Mendelian randomization studies reported no association of genetically predicted self-reported moderate to vigorous physical activity with risk of coronary artery disease, ischemic stroke, peripheral artery disease, or heart failure. The genetic instruments used in these Mendelian randomization studies explain little variation in the physical activity phenotypes, i.e. between 0.1% and 0.2%. Thus, the negative Mendelian randomization findings may reflect insufficient power to detect weak to modest associations. End quote. So, in this example, we have loads of evidence and data from randomized controlled trials and observational studies suggesting that exercise uh, decreases risk of cardiovascular disease. However, we see in some Mendelian randomization data very little association between uh, genetic variants that uh, influence physical activity and cardiovascular disease outcomes. This is likely because those genetic variants have a very small influence on how much physical activity somebody is doing. So, regardless of, or in other words, just the genes themselves don't predict very strongly how much physical activity somebody's going to be doing. So, there's a very very small signal that we're trying to pick up here. This is kind of similar to if we're looking at studies where I've discussed kind of absolute and relative risk. If there's such a small difference between subjects, we're going to need a lot of subjects to pick up the difference, and we still might not pick up the difference, as is the case in this instance. So, exercise, for example, isn't a great candidate for accurate Mendelian randomization findings because genetics don't strongly influence physical activity, at least according to what the authors are saying here. Another example on kind of the other end of the spectrum is LP little a, which is a protein variant on lipoproteins, which increases risk of cardiovascular disease. And it's heavily influenced by genetics. And so we can clearly see in Mendelian randomization studies the same results that we get from randomized controlled trials and observational studies where LP little a increases risk of cardiovascular disease. Quote, the variation in the risk factor explained by genetics is higher for risk factors that are less influenced by environmental factors. As an example, circulating lipoprotein A, LP little a, levels are mainly determined by genetic variations at the LP little a locus. Genetic variants that have been used to proxy the effect of LP little a explain over 60% of the variation in LP little a levels, end quote. So that right there, that last piece is a very important part, or a very important point, in that when it comes to LP little a, the genetic variants influence 60% of the variation, or 60% of the differences between individuals in terms of LP little a levels can be predicted by uh, differences in genetics. Whereas when we looked at exercise, it was only a fraction of 1% could be predicted based on genetic variants. And kind of wrapping up with these requirements, concerns, and limitations, the authors address how Mendelian randomization studies are only one piece of the puzzle. And just like they talked about at the beginning of the study, they're not a panacea. Rather, they're just another piece of evidence to kind of add to an overarching body of evidence to provide more context and more certainty regarding a given outcome or a given field of research. Quote, although Mendelian randomization studies can add an important piece of the puzzle on the possible causal effect of a risk factor on a health outcome, Mendelian randomization findings should be interpreted in the light of evidence from other sources such as traditional observational and experimental studies. As an example, results from prospective cohort studies have shown that high circulating calcium levels are associated with an increased risk of myocardial infarction. Furthermore, a meta-analysis of 3 randomized controlled trials showed that relatively short-term high-dose calcium monotherapy or calcium plus vitamin D supplements, both of which result in slight but significant increases in serum calcium levels, are among the most commonly prescribed among the most commonly prescribed therapeutics, increase the risk of myocardial infarction. On top of this evidence, Mendelian randomization investigations have found that genetically predicted lifelong higher serum calcium levels are associated with an increased risk of coronary artery disease, myocardial infarction, and overall cardiovascular disease. Hence, triangulating the evidence across study design supports a causal association between short-term and lifelong modest elevations in circulating calcium levels and a higher risk of myocardial infarction, end quote. So the key point here, I think, is that Mendelian randomization or the implementation of Mendelian randomization isn't throwing out the idea of kind of triangulating or putting all of the different pieces of the puzzle together from different bodies of evidence and using them to kind of cover each other's cons with each other's pros, like I described in the beginning. Rather, that Mendelian randomization is just an additional tool that we have now to kind of confirm or thwart other findings to increase or decrease our certainty in the probability of an outcome occurring. Now, to close it up, I figure I would address some kind of cool applications beyond what I've already described that the authors kind of talked about towards the end of the paper. One is multivariable Mendelian randomization. And this essentially is the concept of combining data from different variants that affect different risk factors, or rather looking at how taking into account different risk factors in your Mendelian randomization analysis. You can tell I'm not exactly the statistical master when it comes to this stuff. Um, but this is similar to the idea of how in observational studies, we can take other confounding variables into account and put them into our statistical analysis to try to account for them to see how much impact they have. And this can be important in kind of trying to isolate causal variables. Quote, multiple variable Mendelian randomization, a statistical method that allows for the association of genetic variants with multiple risk factors to be incorporated into the analysis. The approach can be used to adjust for known confounders or to explore the mediating effects of factors that are in the causal pathway from the risk factor of interest to the outcome. End quote. Another quote, multivariable Mendelian randomization analysis can also be applied to explore mediating effects of factors that may lie in the causal pathway from the studied risk factor to the outcome. As an example, multivariable Mendelian randomization analysis was performed to evaluate the mediating effects of cardiometabolic risk factors on the association between adiposity and common atherosclerotic cardiovascular diseases. The study showed that genetically predicted systolic blood pressure and type 2 diabetes liability mediated 27% and 41% respectively of the association between genetically predicted BMI and risk of coronary artery disease. Adjustment for blood lipids and smoking liability through multivariable Mendelian randomization analysis resulted in only minor attenuations in the association estimate for genetically predicted BMI in relation to coronary artery disease, suggesting that lipids and smoking were not major mediators or confounders of the relationship. End quote. In this example, the authors are talking about how in a given multivariable Mendelian randomization, we can assess the degree to which different risk factors are involved in increased BMI, increasing risk of cardiovascular disease. In this case, the results showed that systolic blood pressure and type 2 diabetes liability, or as I understand it, risk of getting type 2 diabetes, genetic influence on those two variables predicted a considerable amount of the outcomes the influence that BMI exerts on the outcome of interest, which was atherosclerotic cardiovascular disease. Another example, quote, with respect to circulating lipids, Mendelian randomization studies have concluded that atherogenic lipid-related entities, including LDL cholesterol, apolipoprotein B, and Lp(a) are associated with an increased risk of atherosclerotic cardiovascular diseases, and that genetically predicted Lp(a) levels are associated with atrial fibrillation risk. Multivariable Mendelian randomization analyses have suggested that the associations of LDL-C with risk of coronary artery disease, ischemic stroke, and peripheral artery disease are largely driven by apolipoprotein B. Mendelian randomization findings have not supported the independent causal role of HDL cholesterol in major cardiovascular diseases after accounting for LDL-C and apolipoprotein B, end quote. So in this case, the tool of multivariable Mendelian randomization analyses allows us to get a little bit closer to predicting causality. So although LDL-C is associated, even in these in these Mendelian randomization studies, when it's isolated, it's associated with increased risk of atherosclerotic cardiovascular diseases. But when we account for ApoB, we see that the reason LDL-C is associated with these outcomes is because LDL-C is associated with ApoB. And when we factor in ApoB, we no longer see the association. But when we do the inverse, we still see an association, suggesting that ApoB, even when considering LDL-C, is the causal factor for atherosclerotic cardiovascular disease. And the counter is, or not counter necessarily, but another example is for HDL. So when we factor in ApoB and LDL-C, we see that HDL cholesterol is not predicting major cardiovascular diseases via these Mendelian randomization studies that are taking into account LDL-C and apolipoprotein B. Another interesting application of Mendelian randomization studies comes with drug research, which I kind of alluded to way at the beginning, in that this particular field of research takes a long time and has a lot of ethical considerations when it comes to randomized controlled trials. And also, as I understand it, it isn't observational studies aren't super helpful for drug research as well, and still kind of have a lot of particular holes to be filled, or they really aren't useful or even used, I guess. Mendelian randomization studies, on the other hand, are really useful for particular drugs that target very specific proteins because they're in the case where there are genetic variants that essentially mimic what it would look like in an individual if the drug had its interaction with that protein. We can see first whether or not it's effective over long periods of time for reaching the given outcome, and second, whether or not it will lead to an increase in adverse events. Quote, in brief, most drugs act by targeting proteins which are coded for by genes. Variants in the region of the relevant protein coding gene can thus be used to proxy the pharmacologic effects of perturbing the corresponding drug target. Whereas Mendelian randomization studies of modifiable risk factors generally employ variants from multiple gene regions, Mendelian randomization analyses exploring drug target effects typically utilize variants from a single gene region, specifically variants within the region around the protein coding gene. Such an analysis is known as cis Mendelian randomization analysis, as variants near the protein coding gene are named cis variants. A prerequisite is that the selected genetic variants represent the clinical effects of the drug target. As an example, Mendelian randomization has been applied to predict the effects of cholesterol-lowering drugs that target 3-hydroxy 3-methylglutaryl-CoA reductase, or HMG-CoA reductase, the target of statins, Niemann-Pick C1-like-1, which is a protein that's the target of ezetimibe, another cholesterol-lowering medication, and proprotein convertase subtilisin slash kexin type 9, PCSK9, which is another protein that's the target of PCSK9 inhibitors, another cholesterol-lowering medication, and cholesterol ester transfer protein, or CETP, even though it's spelled CETP, target of CETP inhibitors on cardiovascular disease risk. In one of these initial Mendelian randomization studies, the researchers selected variants within uh, a certain proximity to the HMG-CoA reductase and Niemann-Pick C1-like-1 genes that associated with LDL cholesterol levels at a threshold of less than p less than 5 to the 10 minus a sixth in the Global Lipids Genetics Consortium as instrumental variables to predict the corresponding drug effects. So essentially, each of those medications that were just listed are classic cholesterol-lowering medications that target specific proteins. For example, HMG-CoA reductase is a protein that is or an enzyme that is involved in the rate limiting step of cholesterol synthesis. And so statins, common cholesterol lowering medication, target that enzyme and inhibit it to decrease the amount of cholesterol synthesized in the liver, which then causes the liver to try to pull more cholesterol out of the blood by pulling LDL particles or ApoB particles out of the blood to kind of refill itself or restock itself on cholesterol. Another example is PCSK9, which is a protein that's found in your blood that will attach to ApoB proteins or ApoB particles. And when those ApoB particles, like LDL, are pulled into the liver by the LDL receptor, PCSK9 will stick to the LDL receptor and drag it into a part of the cell where it will be degraded and recycled. And the amount of PCSK9 you have influences how much or how many LDL receptors you have on the surface of the liver, because the more PCSK9, the more degradation of those receptors, the more recycling of those receptors. The less PCSK9, the less recycling of those receptors, the more you have. And the more you have is, in theory, beneficial because you'll be able to pull more ApoB apolipoprotein B carrying lipoproteins like LDL out of your blood, and lowering your LDL is, in theory, beneficial for reducing your risk of atherosclerosis. Anyways, the point of all this is that we can look at genetic variants that impact HMG-CoA reductase and PCSK9. For example, some genetic variants might consist of mutations where an individual is not able to produce PCSK9, or they're only able to produce very little amount of PCSK9, which means that they'll have very little PCSK9 binding to LDL receptors. And like I just described, this will increase the amount of LDL receptors they have in the liver and decrease the amount of low density lipoproteins or ApoB carrying lipoproteins in their blood. So pretty much what they'll do is they'll look at these individuals with these genetic variants, and they'll see whether or not they have increased or decreased or neutral impacts on their risk of cardiovascular disease. And they'll also be able to see whether or not individuals with these genetic variants are at particularly increased risk of other adverse events. And so they can use these to kind of predict whether or not taking a given drug that has a similar or the same effect as that genetic variant does, and assess whether or not that drug is more or less likely to lead to adverse events over a lifetime and more or less likely to lead to the desired outcome over a lifetime. Quote, the analysis showed that genetically predicted LDL-C lowering mediated by variants in the HMG-CoA reductase or Niemann-Pick-C1 like 1 gene or in both genes was associated with a reduced risk of coronary heart disease, end quote. So pretty much the genetic variants that replicate what happens or the effects of statins and ezetimibe, which is the medication that acts on the Niemann, geez, Niemann-Pick-C1 like 1 protein. These names are hard. Both of those genetic variants decrease risk of cardiovascular events. Quote, an important limitation of this approach is that the extent to which genetic variants mimic the action of specific drugs is often unclear. Further limitations are that genetic variants have lifelong effects, whereas trials assess the impact of short-term interventions. Trials often compare the impact of interventions on top of standard care, such as statins, and hence Mendelian randomization investigations may not reflect real-world practice, and trial endpoints often differ from outcomes in Mendelian randomization analyses, end quote. So although Mendelian randomization can fill some of the gaps when it comes to drug research, for example, they also may have some issues with ecological validity, just like randomized controlled trials do, because although we can look at how these variants impact outcomes, in real life, these variants or the drugs that are pretty much mimicking the effects of these variants aren't being taken in isolation. So if we see that taking statins, for example, alone, or the genetic variants that show similar effects to what happens when you take a statin lead to decreased risk, it may not replicate or be exactly predictive of what would happen in real life if someone took a statin plus ezetimibe or plus PCSK9, something like that. Quote, Mendelian randomization has been applied to evaluate the repurposing potential of available drugs. As an example, Mendelian randomization studies have demonstrated that genetic mimicry of interleukin-6 receptor blockade targeted by tocilizumab is associated with lower risk of rheumatoid arthritis, but also with reduced risks of several cardiovascular diseases, as well as COVID-19. Nonetheless, a possible side effect is an increased risk of pneumonia. These findings suggest that blockade of the interleukin-6 signaling pathway may be a target for the prevention of diverse cardiovascular diseases in COVID-19, but that caution should be taken with regard to possible adverse effects, end quote. So this is another kind of interesting application in that Mendelian randomization can help us understand whether or not existing drugs that we're already using for one use case might be useful for another use case, and it can also help us predict whether or not there'll be side effects that we won't want if we do make that kind of exchange, or if we start using that original drug for another use case. And that, I think, is all I have. This has been a long one. I've been, resorting to drinking water because I'm talking so much my throat's getting dry, and I certainly I'm starting to get fatigued. I'm babbling over quotes and stuff, but if you stuck along this whole time, kudos to you. I hope that this was useful. And if you want more similar content like this, if you like this, if you found this useful, I post more stuff like this on YouTube and also on my Substack, Shortcut U. You'll find video content as well as written content that is likely a lot more succinct and organized than when I'm just blabbing off to you, reading quotes and kind of going off on tangents. And if you want to support my work, support Shortcut U, you can subscribe on YouTube and also at Shortcut U. And if you have been following along with several of these Talking Studies posts, feel free to let me know in the comments what you think of them, whether or not you think I should keep doing them, whether or not they've been useful, anything like that. And with that, I will catch you next time.